Welcome all. Today I'm going to speak about lipoprotein metabolism. First, I'm going to start with Kynomicron, then very low, intermediate, and low density lipoproteins, and then end with high density lipoproteins. So catch your breath and get ready. It is about to start, and here we go. First of all, Kynomicron. Kynomicron is the largest and the least dense among all. It is synthesized in the small intestine, consists of B48, a apolipoprotein, along with dietary triglyceride, cholesterol, and phospholipid, and then it moves to the blood. In the blood, Kynomicron meets HDL, and this guy do him a favor by giving him E and C apoproteins. These apoproteins have different functions. At first, C2 apoprotein activate lipoprotein lipase, an enzyme exposed on the endothelial surface of a peripheral tissue. This enzyme destroys triglyceride from chylomicron, turning them into fatty acid and glycerol. Fatty acid is stored in these cells and either used for energy or stored as triglyceride. And the chylomicron now is getting smaller and more dense as it loses lipids. Now this particle is called chylomicron remnant. It's taken up by the liver, but first by the CETP, it exchange the cholesterol ester from HDL in exchange for triglyceride, phospholipid, and C2. Then cholesterol ester passes with chylomicron to the liver, and the chylomicron remnant use the E apoprotein as a ligand for the ApoE receptor, which open the gates for the taking up of chylomicron remnant particle, which is destroyed by the lysosome, which produces amino acid, fatty acid, and free cholesterol. VLDL, very low density lipoprotein. This lipoprotein is synthesized in the liver by a protein called the MTP. MTP protein uses magic to gather triglyceride, cholesterol, and the ApoB100. And this magic produce VLDL. B100, of course, not B48. This VLDL is called nascent VLDL, and it passes from the liver into the blood as nascent VLDL. So it shares kind of the same fate as chylomicron, as it in the blood it also receives a protein E and C2 from the HDL. So it's kind of similar like chylomicron. So except for the B100 instead for B48. And as the very low density lipoprotein passes into the blood, it meets the lipoprotein lipase, which is exposed on the endothelial surface of peripheral tissue. It is activated by the C2, just like in chylomicron. Lipoprotein lipase destroys the triglyceride from the VLDL into fatty acid and glycerol. Fatty acid pass to the peripheral tissue either for storage or for oxidation. And now the VLDL is no more called VLDL. It's called VLDL remnant, or another lipoprotein called intermediate density lipoprotein, as it passed from very low to intermediate. This IDL goes through two pathways. The first, it is either taken up by the liver, as it still contains the ApoE protein, and C2, it passes to the hepatic tissue. The hepatic cells, as we explained in chylomicron, have the ApoE receptor. This receptor accepts E as a ligand and opens the gates for the take-up of the VLDL remnant or IDL, which is destroyed just like chylomicron by the lysosome into amino acid, fatty acid, or free cholesterol. The other pathway of IDL in the blood, it meets the HDL. The HDL is loaded with cholesterol ester. At first, the HDL takes back the apoprotein E and C2 from the IDL, and it contains a protein called CETP, cholesterol ester transfer protein, which exchanges triglyceride from IDL and cholesterol ester from HDL. And now the HDL leaves the scene, and the IDL particle is no more called IDL. It is called LDL, a new lipoprotein. This lipoprotein, after the exchange, has low concentration of triglyceride and high concentration of cholesterol ester. The LDL now is loaded with cholesterol ester, and it's considered the badass among lipoproteins. It passes to the tissues, the liver or the peripheral tissue. The LDL contains still the ApoB100. Now it acts as a ligand for a receptor called ApoE B100 receptor, which open the gates for the take-up of LDL and destruction by the lysosome, releasing large amount of cholesterol and free fatty acid and amino acids. And as I said, the same series of events occur in the peripheral tissues, muscle and adipose tissues. And that was it for LDL. However, there is one more thing. As the concentration of cholesterol increases into the cell, the receptor closes and the LDL build up into the blood, which is removed by macrophage. This results in the transformation of macrophage into foam cell. These cells build up into the endothelial cells on the arteries, causing atherosclerosis. And thank you for watching.